Today we're learning about Xylem and Phloem transport systems, how transpiration drives water movement through plants, how factors like light, temperature, airflow, and humidity affect transpiration rates, and finally, how guard cells control stomata to regulate gas exchange and water loss. Despite what you might imagine, plants don't have tiny vehicles moving substances around inside them, but they do have fascinating transport systems. Let's start with phloem tubes, which transport food throughout the plant. Phloem consists of arrangements of living cells that have specialized end walls with small openings or pores. These openings allow the contents to move from one cell to another. Phloem's primary function is to move food substances, especially the sugars produced in the leaves, to other parts of the plant. This nutrient transport provides energy for growing areas or gets stored for later use. Interestingly, this movement can occur both upward and downward in the plant, depending on where nutrients are needed. The technical term for this movement of food through phloem is translocation. Now let's examine xylem tubes, which are responsible for water transport. Unlike phloem, xylem is composed of non-living cells connected together without separating walls, creating what's essentially a continuous pipeline. These xylem vessels are reinforced with a substance called lignin that provides strength and rigidity to maintain their structure. Xylem transports water containing dissolved minerals from the roots upward to the stems and leaves. This continuous flow of water from roots to leaves is known as the transpiration stream. So, what exactly is transpiration? In simple terms, it's the process by which plants lose water to the atmosphere, but the process is more complex than it first appears. Transpiration occurs through evaporation and diffusion of water from the plant's surfaces. The majority of water loss happens through the leaves via small openings called stomata. When water evaporates from the leaf cells, it creates a mild water deficit, which pulls more water up from other parts of the plant through the xylem vessels. This in turn draws more water up from the roots, creating a continuous flow of water moving through the entire plant. An interesting point to note is that transpiration isn't actually the plant's goal. It's a consequence of the leaf structure needed for photosynthesis. Plants must have stomata to allow carbon dioxide to enter for photosynthesis. However, the concentration gradient means that water inside the leaf will inevitably escape through these same openings. This water loss is a necessary compromise for the plant to carry out photosynthesis. Now let's investigate the factors that influence how quickly plants lose water. Four main variables affect transpiration rates. Light conditions significantly impact transpiration. More intense light leads to increased water loss. This occurs because stomata tend to close in darkness. Since photosynthesis cannot occur without light, plants don't need to keep their stomata open for carbon dioxide intake in the dark. With closed stomata, minimal water escapes from the plant. Environmental temperature also plays a crucial role. Warmer conditions accelerate transpiration. This happens because heat gives water molecules additional energy to evaporate and move out through the stomata. Air movement around the plant is another important factor. Stronger air currents increase water loss. When air is still around a leaf, evaporated water accumulates near the leaf surface. This creates similar water vapor concentrations inside and outside the leaf, slowing further diffusion. However, when air currents remove this moisture layer, the concentration gradient is maintained, allowing continuous, rapid diffusion from high to low concentration. The fourth factor is atmospheric moisture. Drier surrounding air accelerates transpiration. Scientists can measure transpiration rates by monitoring water uptake by plants. This approach works because the amount of water absorbed by the plant correlates directly with water lost through leaf transpiration. To conduct such measurements, researchers use equipment featuring a plant with roots in water, a narrow tube, and a method to track air bubble movement in the tube. By noting the initial position of the bubble and the distance it travels over time, one can determine the rate of water absorption. For accurate results, it's essential to maintain consistent experimental conditions, particularly regarding temperature and humidity levels. Let's now examine guard cells, which regulate stomatal openings. These specialized cells have a distinctive curved shape that enables them to control the size of the stomatal pore. When guard cells absorb water, they become swollen and firm. This change in shape creates an opening between them for gas exchange during photosynthesis. During water shortages, guard cells release water and become less rigid, causing the stomata to close. This mechanism helps prevent excessive moisture loss from the plant. The ability of guard cells to change shape comes from their unique structure. They feature thinner walls on their outer sides and thicker walls on their inner surfaces, which influences how they deform when their water content changes. In conclusion, plants have evolved remarkable transport mechanisms for moving water, minerals, and nutrients to where they're needed.